Well, hi everyone, and welcome to today's Seed World Canada Retail Strategy webinar. My name is Mark Zinkowitz, and I serve as editor for Seed World Canada. And I'm happy to be your host. Today is a special edition of our webinar. We're featuring our three recent Canadian plant breeding innovation scholars. We're going to talk about their research, what makes it so special, and how seed growers and retailers could benefit from us. Hello, everyone. Good to see you. How are you today?
Great, Mark.
I'm all good to go.
Yeah.
Thank you.
Good, good. Good to see all of you. First, I just want to thank our sponsor, CCAN, for their support. If you have a question during the webinar, please type it into the chat box at any time. You don't have to wait until the end of the webinar. If question pops into your mind, please just type it into the chat box and let us know who. If it's directed at a specific person, just let us know who specifically your question is directed to. We will be posting a recording of this webinar on our website, seedworld.com/Canada, in the coming hours. So this webinar is being recorded, and you'll be able to view it on demand. Like I say, we are profiling our three Canadian plant breeding innovation scholars for 2025. I want to acknowledge the sponsors of the Canadian Plant Breeding Innovation Scholarships. CCAN, of course, is one. We also have Alberta Grains, Canadian Seed Growers Association, FP Genetics, High Tech Production, Richardson, Sasqueat, Seeds Canada, Seed World Canada, Warburton's, and the Western Grains Research Foundation. So thank you to all of our wonderful sponsors for making these scholarships possible. And like I say, we have our three scholarship winners we've read about on our website and in our January 2025 edition of Seed World Canada. We have Isabella Charvelati from McGill University. Did I pronounce that right, Isabella?
Yeah, well done.
Perfect, perfect. We have Quinn Sturby from the University of Saskatchewan. Welcome, Quinn. And Jared Bento from the University of Manitoba. I'm also thrilled to have Todd Hyra online with us. He is CCAN's Western Business Manager. And Todd's going to break down why the cutting edge research that we're about to explore is really a game-changing research for the seed sector. So welcome, Todd. Thank you for joining us.
Thanks, Mark.
And thank you, Quinn and Isabella and Jared, for joining us. I'm going to go in alphabetical order today and talk with each of you. And so, Jared, I'm going to highlight you on our virtual stage. So we will touch base with Todd and Isabella and Quinn shortly. Jared, again, thank you for your time today. And we're going to talk about some of this really cool research that you're doing. Now, you're using DNA data and artificial intelligence to help breed better canola faster. And I know your research is like there's so much to it. It's really multifaceted. But one part that really stood out to me that instead of relying, instead of relying only on traditional field trials, which can take years, as we know, you're testing computer models that can predict important traits like oil content and fatty acid composition. I'm curious, how did you get involved in doing this research? Was there a particular challenge or opportunity in the industry that drew you in? How did that all kind of start out for you?
Well, first, just to start off, thanks for having me here, Mark and Todd. I'm really glad to be able to talk about this with you alongside Quinn and Isabella. So regarding getting into this research, it wasn't really a project that I proposed myself specifically, and I thought that, I mean, like many of us are just bachelor students that want to get into research, and so we find a project that is kind of given to us. So this research project was very important to my supervisor, Dr. Duncan, and he proposed that it could be a good project for me to get into. So once I got to learn more about it, I realized that it was actually very much related to me, because you say it's important to reduce our reliance on traditional field trials. I found it important because I was one of those people in the field trials, not coordinating them, but my boots were on the ground. I had the notepad and the pen and paper, walking up and down rows, blocks, day in, day out, taking notes on people's blocks that, I mean, I had no idea where exactly the data was going, but I knew that the data was important for making these selections and seeing how real varieties are behaving in the environment. So this, I would say, is what kind of kept me coming back to my own research. Staying interested, knowing that it has a real world impact not only on the breeders themselves that coordinate these field trials, but also people like me who have their boots full to the brim with well, Manitoban clay, but also, I mean, anyone in Saskatchewan and Alberta can attest to that feeling walking up and down their blocks. So that's what
Yeah.
kind of kept me coming back.
Well, and field trials, of course, I mean, they're such a necessary part of our industry and probably always will be. But like you say, when you're the boots on the ground, when you're the one who's got to do the work and be out there, like it's hard work out in the field. And so it's like, yeah, it's probably like, geez, if there's any opportunity here to kind of make things maybe a little bit easier on ourselves, hey, we should definitely explore it. And so I'm gonna kind of go through some of these slides that you sent me. We're looking at the bioinformatics tool belt. And so feel free to kind of fill us in as we go and explain what we're kind of looking at here. But your work supports the rapeseed and canola breeding program at the University of Manitoba. And so, yeah, I'm just curious how your research fits in to the bigger picture of breeding, not just canola, but better varieties in general. Can you kind of walk us through a little bit of what we're looking at here and just explain kind of how this all works?
For sure. So I'm glad that you mentioned that it's not just for canola, it's not just for oil and anything. The interesting thing about GS's genomic selection is that it is a selection method that is applicable to so many traits and to so many species. As long as you pretty much have the genotypic data and some pre-existing field data, GS is very much applicable. It doesn't really depend only on the complexity of the trait. And so, actually, if you want to take a look at one of the next slides, I think the filter slides. Um, so this is a big picture that I like to look at a lot whenever I'm describing GS, looking first at the context of conventional selection. So this is how breeding has been done for hundreds of years, even thousands if you consider, plant improvement just passively. But every single year of plant breeding requires hundreds, if not thousands, of individual genotypes replicated across many different locations. You see the map in the second panel, and obviously lots of man hours and also carbon input, carbon emissions for 1 year, for 2 years, for 3 years. Many breeding programs to go from an initial hybrid or initial cross to a cultivated variety released to farmers can take up to 10, 15 years. And so when you compile all of that input, it definitely adds up. Can be millions and millions of dollars, hundreds of thousands of tons of carbon emissions. So obviously anyone can see where we want to go, and that's less. We want to use less, we want to be more sustainable. So if you actually go to the next slide for this, breeding acts as a filter. Essentially we have a wide population and we want to whittle it down to the best genetics available in the population. And that's not easy. A lot of selections need to be made, and it all depends really on your selection intensity, your accuracy, the time of generation, just to move that population and the alleles, the genes available in that population towards a trait, towards a phenotype that is desirable. So what GS does is helps to narrow that filter a little bit quicker. I have it again in the next slide, just to take. Oh, I actually didn't have that filter, but basically it has that filter and helps to increase our selection intensity and increase the accuracy without sacrificing the time that goes into it. So basically, if we increase our intensity and we increase our accuracy, we can increase our genetic gain over time. And this, of course, is found in the breeder's equation. It's something that many of us in plant breeding see pretty much first day, but for anyone who hasn't seen this equation before, basically we want to have our selections as accurate and as intense as possible to reduce that filter, or to use the filter and move towards a desirable product. So if we could actually take one last quick look at the first picture, the kind of workflow of this. It can be applicable to any trait, to any crop, basically using pre-existing data in a training population, combining that genotypic and field data to train a computer model, whether it be machine learning or any other type of computer model. And then we can incorporate just the genetic data of hybrids that we're interested in. So these are things that we don't have to put in the field. They don't need to be replicated hundreds of times over 15 years. We can plant one plant in a greenhouse, take some genotypic data from the leaf, and then use that trained model to predict its traits across a population of, let's say, 500. And then using that trained model, we can now make our selections without ever having to put those plants into the field. It's not an end-all be-all, but it's a great tool for breeders to be able to reduce their reliance on testing hundreds and hundreds of genotypes that would never actually reach the end of the filter anyways. So that was a pretty long-winded answer. I didn't mean to do that, but yeah, it fits into the bigger picture of breeding very much so.
Yeah, no, not long-winded at all. I mean, these things take some explanation. They're definitely not simple. And I know this is just, it's in the research phase, but I'm just curious, like, are you already seeing results that might hint at how your methods could speed up the development of improved crop varieties? What can you tell us about kind of what you've discovered so far?
Definitely. So in my own research, I've seen many projects in the past focused on not only on canola, but different important crops. Soybean, corn specifically is very heavily researched in GS. And we see some results where breeding programs can be cut down by 3 years, or 1 year even. 1 year, it doesn't sound like very much, but when you consider the amount of resources that I showed, it stacks up very heavily. And so just to reduce 1 year is thousands of tons of emissions and dollars. And not only that, but if a challenge can be identified by a plant breeder or an organization, and it can be addressed by a new cultivar just 1 year earlier for farmers, I mean, I don't have to tell you how important that is if we can get ahead of a disease and save 1 whole year of yield against a disease that is emerging. Verticillium stripe rust comes to mind. That is a very much looming threat for canola farmers across Canada. It is already destroying massive majorities of yield, and it is present across many fields. So that is one thing that we keep in mind for, getting ahead of it and using methods like this to address these issues as early as possible.
I'm just going to bring Todd Hyra to our stage here so that Todd can kind of chime in a little bit. And then Todd, I just wanted to get your take on this. I'm just curious about this concept of reducing reliance on field trials and using computer models to predict traits. Like I said at the beginning, field trials are, the gold standard, probably always will be, but it's exciting to have a tool that, could help the process along. I mean, the more tools we have, the better, right? And I'm just curious what potential you see here for SeedCann's customers and for seed growers and seed retailers in general.
Yeah, thanks. I think Jared's done a really nice job of outlining the potential here. So first of all, say hi to your supervisor. Rob Duncan was a summer student of mine many years ago, back when I was in research. So full circle here. I like your funnel analogy, and it's about making what goes into the top of that funnel better and more useful. And so you're not wasting time, energy, resources on something that's going to be throwing away. As you're going through that funnel process, and each year you're whittling away, trying to find the best of the best. And if you can discard products that are of no interest off the start before you're wasting resources in the field, which is a more costly part of screening, then you're making the entire process better. Our customers, our seed growers, and ultimately our farmer customers they don't want more, they want better. And so, as you said, if you can get something to them a year or two faster, it's a big deal. But they always want them to be better than what they've had before and solve a problem that they're dealing with in the field. And so, that making that funnel more useful, it's a very valuable tool.
Yeah, well, and that's what I think defines Canadian plant breeding innovation scholar, right? Is the fact that you're making things better, and that's key for our industry. I mean, without bright young minds, we can't do that. We need new people with new ideas, and so yeah, this is super exciting indeed. Thanks, Todd, and thank you, Jared, for your insights. Hang on the line, don't go anywhere. We'll be doing a Q&A at the end. I'm going to talk to Isabella next, so hang on the line. Hi, Isabella. How are you today?
Hey, Mark, I'm doing well. How are you?
Good. I am very well, and I'm a little bit warmer than I've been in a while because it's warming up here in Manitoba, and so that is always good news. No more minus 30. Thanks for joining us, Isabella. And your research is really exciting. I'm just going to explain a little bit here about what you do. You're exploring how predictive models impact breeding success over time. Your PhD research focuses on bridging that gap between traditional breeding methods and genomics assisted selection, which provides breeders with, you're providing them with a simulation platform to navigate this transition. And so I'm curious, a simulation platform, that sounds really cool. Can you talk a little bit about what this is? What does this look like, this simulation platform? Let's say I'm a breeder, I'm a researcher, and I want to use this platform. What does that look like? How does it work exactly?
Mm-hmm. Yeah, that's a good question. First of all, it's great to be here with Jared and Quinn and Todd. So thanks for having all of us. And Jared did such a great job at outlining what genomic selection looks like. And we can get a sense from all the information he gave us that this is not simple. There are a ton of moving parts. So if you're a breeder and you're looking to start implementing some genomics methods, you have a ton of decisions to make and your valuable resources are at stake. So that's where the simulations come in. So if we're thinking about genomic selection, we have to think about what is our training population going to look like? At what stage in our breeding program do we want to start using a prediction model? Do we want to be using a model to select parents, or do we want to be using a model to evaluate lines and reduce the volume of our field trials? So there's all these questions. So for the simulations, we can run all of these different scenarios and get a sense of what the outcome will be before we take it to the field. So what we would do is just use a few simple commands in the computer to outline what our breeding program looks like. What does our crossing block look like? Are we selfing? How many crosses are we making? How many generations? Everything like that. You can just type it in, run as many breeding cycles as you want, and then get a sense of what your gains will look like, how the diversity in your program is impacted, and we can use that to guide our work in the field.
So speaking of the field, I've got a few slides from you here, Isabella, and what are we looking at here?
So this was last summer. We are doing an increase, a seed bulk, to get ready for our field trial that we're doing this summer that we're very, very excited for. And the cool thing about the plants that you see here, these are cranberry beans. These are progeny of parents that we selected using a prediction model. So when we initiate a new breeding cycle, we start by crossing parents, and selecting those parents is really important. And we used a prediction model just like the ones that Jared described to us to identify top parents. And the idea is that the model lets us select directly on the genotypes that are ideal for us instead of kind of indirectly selecting on genotypes based on the observed phenotype. So we have these genomically selected parents, we cross them, we advance them, and here they're just bulking up seed to test them in the field next summer.
Ah, neat. Okay, so you kind of these must be the beans here, I'm thinking.
Yes, exactly.
Ah, cool. Okay. And so, like, in terms of the long-term insights from your predictive breeding models, like, this is something that, and we'll get Todd's take on this shortly, but this is something I think could really empower seed retailers to better communicate, the value of genomics assisted breeding to farmers. And so for people who grow seed, who sell seed to farmers, like, can you speculate a little bit on, how this could really improve that whole process?
Yeah, for sure. I think there's really two main points. The first one is accuracy, and the second one is speed. So in terms of accuracy, like genomic selection is really great for traits that have a complex genetic architecture, and those are really important traits like yield or disease resistance. So I think if breeders and retailers are communicating to identify specific goals that target these complex traits, we're going to see greater quality over time much faster. And also, we're living in a very, very fastly changing environment. Like ecologies are changing, economies are changing. And if we're going to be moving fast, we want to make sure that we're moving fast in the right direction. So I think I would just encourage seed retailers and breeders to just communicate and make sure that we are moving fast towards the right target.
Yeah, I was going to say, like, this is coming. I mean, not overnight, but this is going to really speed things up. I mean, we're looking at a day down the road where new varieties are, going to be coming out a lot faster than anybody's ever known. And we're looking at some plants from one of your greenhouses here that like, this is real. Like, you're already growing plants, crops from this. And so what can, seed growers, retailers, what can they do to really prepare for this? Is there something they should know, or a certain information resource, or something that they can kind of tune into to kind of know what might be coming down the pipe, something like that?
Yeah. Well, I feel like our best resources are really each other. It's important to just put time on your calendar to communicate your goals and your needs. Like, new needs arise all the time. Like Jared had mentioned, like some disease pressure looming. Like, we need to be all on the same page in terms of where we want to move. And I think it's also important to remember that fundamentally, these technologies are no different from traditional breeding. Like, the goal is the same: to identify the best genotypes for our needs and to get these new varieties to seed retailers and to farmers. So although it's an exciting new technology, at the end of the day, the goal is the same, and we can keep that in mind.
Yeah, the basic principles are the same, right? And we're just kind of maybe sort of hitting the fast forward button in some ways, but those basic breeding principles remain intact. It's just as we learn more over time, we learn a few, shortcuts. Hey, we could do this better. We can do this quicker. And so that really helps everybody. I'm going to bring Todd back to the stage and get Todd's take on this. And Todd, I'm curious, as seed development looks poised to get even faster in the coming years, I mean, what role, people like you and retailers, your customers, seed growers can bring in bridging the gap between research like this and actual on-farm adoption of improved seed varieties? What are your thoughts on that? Oh, you might be muted still. Hold on.
There, might have been me.
There we go.
It's pretty cool. The technology and this system will only improve upon itself. And so as the concepts are unveiled and then tested in the field, and then that is used to improve them, it will only accelerate, as Isabella says. And I think the key piece here, she nailed it. It's the it's being in touch with farmers and retailers and what their needs are, what is holding them back. And if I think back 15, 20 years ago, it was plant stature, which I'll use a wheat example, where wheat was always kind of one height. And the introduction of a semi-dwarf wheat that allowed more efficient harvest, faster harvest, less fuel usage, all speeded things up. And so having those understandings of one of the, whether it's a disease or an agronomic limitation, and being able to incorporate those into breeding programs faster, more efficiently, are just going to make things better ultimately for the farm customer down the road. And so these are, once again, all tools that just make plant breeding more efficient, and putting the products into the top of those funnels and running that funnel faster is always going to, it's amazing what's coming down the pipe.
Yeah, well, and your job is kind of getting a little bit harder, Todd, because there's going to be all these new varieties, more varieties coming faster, and then you have to decide at CCAN, hey, which of these, varieties we want to bring to our customers. So you've got your work cut out for you.
Not more, better. We just
better. Yes, that's true. I should. Yes, better. That is a key distinction. Absolutely. Thank you, Todd. Thank you, Isabella, and we will rejoin you shortly. I am going to ask Quinn to come to our stage. Hello, Quinn. How are you today?
I'm good. How are you?
Good, good. Thank you for joining us, and congrats on the scholarship. And I want to highlight some of the research you're doing. You're using UAV-based digital imaging to model vegetation growth and predict yields. And you're really bringing this cutting-edge technology to Canadian breeding programs. Now, your research in digital phenotyping is focusing on capturing growth patterns and canopy measurements in common beans. And what this is doing, this is allowing the creation of this data-driven yield prediction that we've been talking about, that really improves efficiency and precision. And I know I've really simplified it there, trying to put it in layman's terms, but that's sort of the gist of what you're doing. And you kind of explain this to us a little bit, and what some of your findings have been thus far.
Yeah. It's great to be here with you and Todd and the other scholars, Isabella and Jared. I'm starting to see kind of a lot of similarities in our projects and in the goals of our projects, because like them, really what I'm trying to do is, reduce like the time plants spend in breeding programs, get plants and varieties to the market faster. And how we're doing that is, like you said, through digital phenotyping, through measuring the canopy growth. So measuring things like canopy height, area, and volume, but then also measuring their growth rates and how that relates to plant productivity, yield, harvest index, and important things like that. So yeah, notable findings so far. One is just being able to measure those things. You can see in the picture on the screen, beans grow kind of unruly or maybe sporadically. I like to make the comparison to things like wheat or corn or even soybeans, where when they're in a plot, they grow very uniform and they almost have like a flat plane across the top. So when a drone is looking from essentially the top down, it's very easy to get an accurate measurement when the plants, or in the case of the drone, like the pixels, are all at the same height and looking the same. Because in common beans, those vines and those leaves that are sticking out can skew the data. So we do have accurate data. I was able to get accurate data, and I was able to get strong correlations between what I measured on the ground and what the drone was able to measure. So kind of like what Jared said, we are able to rely on things other than, students in the field taking those measurements. And then, so also the other part of my project is a little bit of genetics work. And most interestingly, I think anyways, is that we were able to find QTLs for traits measured by the drone and measured manually in the field within the same regions of the genome.
Cool. And is that what we're looking at here? I really love the colors here, but you've got, I assume these are different like plots of beans and then numbers corresponding to what is that the type of bean, or can you kind of shed some light on this?
Yeah, so this is a orthomosaic that we got from the drone. So the drone takes many overlapping images and then it gets stitched into one larger image. And while this looks, maybe like a 2D picture, there is a lot of data kind of hidden within this image. And so this is just a true color picture of my field trial and of the plots from top down. The white squares or bounding boxes are just to contain all information within that box to that plot, and then the numbers are just the corresponding plot numbers.
Yeah, it looks like you've done like some of these slides here. I know there's obviously you're doing some irrigation here, but it's pretty significant. Like some of the sizes of these plots is really impressive. And I'm curious, like in terms of the role that these image-derived canopy measurements and yield prediction models can play in shaping the future of seed selection. I mean, that's what we're talking about here today. People like Todd who get to offer these better varieties to farmers. And can you speculate a little bit on, like what your research might mean for them down the road?
Yeah, I think it will be huge in seed selection. Really, I'm focused on getting like more information and better information to breeders so they can make more informed selections, and then also hopefully select earlier in their programs and then get those seeds to the market faster. And then we will know, like there'll just be so much more data about what's going to market. We can test more lines in more locations and then hopefully offer, more accurate or precise recommendations to farmers based on that.
Yeah, like I was going to say, like, are there other than common bean, like are there other crops? Yeah, this could improve. I imagine there's other crops that this technology, like we've been talking about, can be used in.
Yeah, definitely. What I'm doing can really, I think, be applied to many crops. I think the interesting thing about doing it in common bean is that their growth and their growth habits are hard to characterize because they have both determinate and indeterminate growth. So I think for sure it would be interesting to see this applied to other pulse crops who maybe grow similar to common bean with maybe a bit of a weird growth habit. But I think this could be definitely applied to many crops.
Yeah, I was gonna say that looks like a really big bag of beans there. So once they start growing, I mean, it's you have your work cut out for you just getting around the field. But no, I'm gonna bring Todd back here to the stage and get Todd's take on this. I had one more slide here I wanted to bring up that showed you doing some of your work with bean here in the field. And Todd, if you'll indulge me for another moment here, Quinn's work highlights, what we've been talking about since the start of the webinar, the power of precision agriculture and breeding. And from your perspective, like how important is Quinn's research in terms of these techniques for really maintaining Canada's competitiveness in global seed development? Because obviously this is something that benefits us at the local level, but I know we've been talking a lot recently in the media about trade and things like that. And I would think that Quinn's work is something that could really highlight Canada from a global perspective.
Well, it's important that Canada has the ability to stay current with all the technologies, all of the and be at or above anyone else. So there's other countries are all competing to develop their own products as quickly as they can, but we need to ensure that we're developing products specifically for Western or Canadian conditions, regardless whether it's East or West. And having access to technology like this that provides precision in research helps move that along quicker. Each one of these each of the three presenters all provide tools to plant breeders to make the the process better. And so whether it's genomic selection or a streamlined system or accuracy in the field, digital readings are going to be more accurate than people in many ways. If you're having a bad day, or if you're having technicians rating things different way, machines don't do that. They're always going to be consistent. And so that consistency, and then related back to actual field results, is just going to once again continue to improve the system. So it's again another powerful tool that makes the breeding programs better, stronger, resulting in better products for farmers.
Yeah, well, and that's a whole other topic in and of itself: humans versus machines, and what can we use machines to do? Like you say, that people can make errors and things, and so if we have machine technology, machine learning, that can really improve things for everyone. So that might even have to be the next webinar. I'm making a note of that right now. Thanks for that, Todd. And I'm going to bring our other scholarship recipients back to the stage for a short Q&A here. If you have a question, like I said earlier, please just type it into the chat box at any time and let us know who it's directed to. Jared, I had one that was directed specifically for you, Jared, and they asked, does this project aim to determine QTLs in plants?
So for those at home, QTL stands for quantitative trait loci. That's a locus in the genome that is directly correlated to association in any trait, whatever it might be, a quantitative trait. So genomic selection is not for identifying QTLs, but it utilizes markers, and it will train, like I said, it will take previous existing field data, combine it with genetic data, and train those models to see how all of our markers across the whole genome affect variants in our traits. So your output from genomic selection, or however you might describe it, doesn't identify QTLs, but it uses, you could say QTLs, it uses markers that are associated to traits to predict the outcome, to predict the phenotype of any plant that you have in your breeding program. So yeah, that's what I would say. And I think Isabella could also chime in on that as well.
Mm-hmm. Yeah, I think that's exactly right, Jared. The nice thing about using a prediction model is that we don't have to identify QTL. We rely on the statistical model to calculate effects, and then we pinpoint genotypes that have favorable effects. So yeah, like you said, QTLs are involved, but we don't have to pinpoint them one by one.
Question regarding limitations. Are there any limitations on freedom to operate with use of these technologies? And this is a question for any panelist. And yeah, I'm curious. I mean, there's always hesitancy with new technologies. I mean, that's something that our industry knows all too well. Is there any limitations on freedom to operate? Have any of you ever encountered difficulty, or do you see there maybe being any difficulty in terms of the regulations surrounding some of the types of technologies that you're using? I will open the floor if anyone wishes to chime in.
I think, Isabella, do you have something to say? I have something to leave off on for this question, but if you have anything, I'd offer you go first.
Yeah, in terms of limitations, I think it's important to remember that, again, this is a specific tool that's ideal in a really specific circumstance. So it's not by any means a silver bullet to make breeding really, really fast and efficient. So in terms of its implementation, of course, there are limitations, and there are cases where it's ideal and cases where it's not. And I think in terms of technologies that involve machine learning and these really big training populations, I think the trickiest point sort of becomes like, intellectual property. We have people who are like writing algorithms for machine learning, or maybe they want to or like keep their training data as their own like intellectual property. That's something that I've encountered. But in terms of like the breeding and seed growing community, I think for the most part, people are willing to collaborate and share their data and share their code and share their model. So that is a smaller limitation.
How about you, Quinn? Have you ever, when you describe your research to someone, or I know this webinar is like, we're not talking about like GMO or gene editing specifically, but I know I've interviewed researchers who work in that area in the past, and there's always public, concern, misunderstandings about those technologies. And for you, Quinn, when you're talking to research about someone, with someone, have you ever encountered any questions of like, well, what's that, or how does that affect our food, or anything like that?
A lot of what I hear from people with the use of drones is quite positive. People are usually very interested in the technology and what it can do for plant breeding and for farmers. But I have talked to people, and I do know that when you're actually out in the field flying these things around, people will stop you, and they're a little skeptical or have questions about why you're doing that, why you're near their farm or whatever. So I think as far as, limitations on freedom in that sense, there might be a little bit, but I honestly haven't, like I said, it's being received quite positively. Practically, there are limitations. You have to have a license. There's like restricted no-fly zones and stuff like that. But for the most part, when you're, out in the middle of the prairies in someone's field, or at a research site or things like that, they're usually not an issue, or there are ways to, get around them with, like I said, with the proper licensing and stuff like that.
Yeah. Jared, you said you had something you wanted to finish off with.
For sure. I think Isabella hit the nail on the head regarding like freedom to use, and like the freedom of data shared between researchers. It definitely is. It's a sprouting technology. It's a sprouting method, and people are free to hold on to their data or share it. Obviously, it's in our best interest, I think, to share our methods and optimize our models the best. But I also noticed there was one part of that question that regarded limitations in this. For my model, or my research specifically, focused entirely on the limitations of GS. So basically taking our population and seeing in what circumstances and what scenarios do our models fall apart, what scenarios do they give us very bad results. And I think that research like that is very important to plant breeders, because we can say that this GS is an amazing tool for sure, but like Isabella said, it is not a silver bullet. It is not something that any researcher can just pick up, plug and play, and it works perfectly. There are many different things need to go right, and only one of them needs to go wrong for us to have very bad results. And so that is something that needs to be kept in mind, and have lots of very capable technicians and professors and breeders in play to be able to make sure that we're using this correctly. And the whole point is to increase our efficiency and our sustainability in breeding. So if we don't maintain that, then we're kind of shooting ourselves in the foot with our silver bullet. So yeah.
Thanks, Jared. I have one question here regarding what type of molecular marker platforms do you find most effective for genomic prediction? Millions of tags or low density, but which are practically usable directly? I have to admit, this one goes a little bit over my head. I am a journalist, not a scientist, but if you're able to shed some light on that, that would be terrific.
I can share what we're using in our experiment. So we're using SNP markers that are genotyped using a SNP chip, and it's relatively high density, 12,000 markers. And this was a really big decision for this project, like what genotyping platform to use. And we chose the SNP chip because it genotypes on 12,000 specific markers. And when you want to train a model and then use it, your test samples need to have those same exact markers because the markers are the features of your model. So something like that, where every time you genotype, you're going to be getting a lot of overlap and a lot of repeatability in your genotypes is really useful.
Yeah,
Same
it's like
here.
you're
Same
working.
here as well.
Sorry to interrupt. Go ahead, Jared.
I just want to say same thing here for canola. We used the SNP chip. It's called the Brassica 60K SNP microarray. And so same thing, reproducibility, maintaining the same markers in your training as you use in the actual application of the model is very important. But I think I saw something, yeah, high density GBS was suggested. It has been used before, and obviously it gives you much more data, much more look into less common polymorphisms in the genotype or in the genome. But I mean, it obviously also increases the resources that you require for maintaining all that data, because GBS, whole genome sequencing, exponentially more data than SNPs. And from my own experience, and I believe probably from Isabella's as well, SNP markers are still genome-wide, and we are able to see very promising prediction accuracy just based on those types of markers.
Yeah, I imagine, Quinn, you have much the same challenge. The more data you generate, the more data there is to wade through. So it must keep you on your toes, I'm thinking.
Yeah, for sure it does.
Yeah, I can't even imagine. But no, thank you for the question. And yeah, thank you, everyone. Like I say, we're going to be posting this on our website. And so you'll be able to watch it again. And if you have a question, we'll also have contact information as well on there for our scholarship recipients. You can email them. If you have questions, you're interested in their research, by all means, please reach out. I had not a question, but more of a comment here from Blair Golday. Mentions Sask Wheat is proud to be a co-sponsor of the 2025 Canadian Plant Breeding Innovation Scholarships and supporting the next generation of bright minds. Great job, scholarship recipients. And yes, great job, Quinn and Isabella and Jared. And congratulations on your scholarships again. And thank you, Blair, for the nice comment. And like I say, a recording of this webinar will be made available to everyone in the coming hours. I want to thank our audience for joining us today, for spending your lunch hour with us. Thank you, Todd, for taking the time to give your insights today. I want to thank our webinar sponsor, CCAN, once again. And again, just recognize our sponsors for the scholarships: Western Grains Research Foundation, Warburton's, Seed World Canada, Seeds Canada, CCAN, Sask Wheat, Richardson High Tech Production, FP Genetics, Canadian Seed Growers Association, and Alberta Grains. And Quinn and Jared and Isabella, congratulations again on your scholarships. Thank you for joining us. We're looking forward to seeing where your research goes in the future. And it's an industry where everybody tends to know everybody else. So looking forward to seeing you pop up in the future again. Thank you so much, everybody.
Thank you.
Thank you.
Thank you, Mark.
Thank you. And thanks again to our audience. Have a great day, everyone. This is Mark Zinkowitz of Seed World signing off.